DNA dependent RNA polymerase is there. DNA dependent RNA polymerase enzyme. This DNA dependent RNA polymerase enzyme, it catalyzes, okay, transcription of all types of RNA, catalytic reactions, okay, in the bacterial cell. RNA polymerase, it binds to promoter region. It binds to promoter region and initiates transcription. What it does? Initiates the transcription. Okay. And initiation of, it uses nucleoside triphosphates as a substrate. Here you can see in the initiation process, okay, RNA polymerase, okay, at the promoter region it is fixed and starting the process. Here, when the initiation started, here you can see elongation of the genetic material, and after that the RNA polymerase and RHO factor both are terminated at the terminator end, and transcription process was done in the bacteria. Finally, polymerase in template depend upon the following complementarity rules. It somehow facilitates opening of the helix, okay, and continuous elongation. Opening of the helix and continuous elongation. Only a short stretch of RNA, if you carefully see, remain bound to the enzymes. In case of polymerase reaches the terminator region, okay, the nascent RNA, whatever it is there, it falls off. So also the RNA polymerase, this result in termination of transcription. When it fall off, RNA polymerase, when it fall off, this results in the termination or stopping of transcription process. Okay, transcription process is terminated or stopped. An initiating question is that how RNA polymerase able to catalyze all three steps, which are, what are the three steps, which are nothing but initiation, elongation and termination. So during initiation process, RNA polymerase is only a capable enzyme, okay, catalyzing the process of elongation of the genetic material or DNA. After this, it associates transiently with initiation factor, with the initiation factor. The initiation factor is shown by this symbol, and the termination factor is shown by this symbol, and respectively association with these factors, association or co-association with these factors alter the specificity of the RNA polymerase. Okay, so it changes the nature of the RNA polymerase. So to either during initiation process or termination process. The nature of RNA polymerase during initiation will be changed from the during termination process. Okay. Finally, in bacterial cells, since messenger RNA does not require any processing to become active, automatically it will be activated, and transcription and translation takes place in the same area. There is no separation of cytosol and the nucleus in the bacteria. Why? Because genetic material is dispersed in the cytoplasm of the bacteria. Many times translation, if you carefully see, this translation whatever is there can begin much before the mRNA is fully transcribed. Here consequently transcription and translation can be coupled in bacteria. But whereas in eukaryotes, if you carefully see the eukaryotes, here there are two types of additional complexities. There are at least three RNA polymerases in the nucleus. What are the RNA polymerases in the nucleus? These RNA polymerases are which including, in addition to the RNA polymerase found in the organelles, there is a clear-cut division. There is a clear-cut division of labor. There is a clear-cut division of labor. The RNA polymerase 1 transcribes rRNA, okay, ribosomal RNA. Now you can see in the diagram process of transcription in eukaryotes. Capping, during capping process at the 5th end, you can see here, and after capping you can see the exons, okay, introns, and at the third sequence mRNA was there, RNA splicing was occurring here, you can see, and polyadenylation process was occurred, polyadenylation, and if you see here messenger RNA at 5th end, okay, and it will be going into the 3rd end, okay. Transcription was moving from one end to another end, and after this, whereas the RNA polymerase 3, if you carefully see, RNA polymerase 3 is responsible for transcription. It is responsible for transcription of tRNA, okay, and whereas 5sRNA, a type of sRNA, and snRNA, small nuclear RNAs, okay, and the RNA polymerase 2, if you carefully see, the RNA polymerase 2 transcribes precursor of mRNA, the heterogeneous nuclear RNA, hnRNA, heterogeneous. Second complexity is the primary transcription. These primary transcripts contain both the exon and introns. In the diagram I have shown you, and which are non-functional. Hence it is subjected to process called as splicing. They are non-functional but helps in the process of splicing, okay, where the introns are removed and exons are joined in a defined order. Heterogeneous RNA, hnRNA, undergoes additional processing called capping and tailing. During capping and tailing, what happens? Tail of the DNA and RNA will be formed. In capping, an unusual nucleotide, in the form of unusual nucleotide, capping occurs. Okay, this unusual nucleotide is made up of methyl guanosine triphosphate. Methyl guanosine triphosphate is there. Okay, it is added to the 5th end of RNA, hnRNA. In tailing, adenylate residues which are added at 3rd in the template independent manner. So it is fully processed hnRNA or heterogeneous RNA. Now it is called as a mRNA. What is that called? mRNA. That is transcribed out and the nucleus is undergoes for translation. Finally, the significance of such a complexity is now beginning to understand. The split gene arrangement, if you carefully see, represent probably an ancient feature of the genome. Split gene is representing ancient genome. The presence of intron is a reminiscent of antiquity, and the process of splicing represents dominancy of a RNA world or RNA as a genetic material. This splicing only occurs in the RNA. In recent times, understanding of RNA is become more and more easy, and it is a dependent process in the living systems or living organisms.